Why do we call this smooth muscle? Very simple. Smooth muscles do, under light microscopy or polarized microscopy, do not show any striations. So we call them smooth. Where do we find these smooth muscle cells? Well, mostly in the wall of hollow organs. But you will also find it in your eye, your pupil, and some other places. Okay? Smooth muscles are usually subdivided at first. In contrast to skeletal muscle, smooth muscles are single nucleated cells. Okay, so these are normal cells with one nucleus. Smooth muscle as a tissue is usually divided into two big groups. One is called the multi-unit smooth muscle. The other one is called the unitary smooth muscle. And this simply refers to the fact of how they are activated and how do they respond to this activation. Multi-unit smooth muscle cells, on the left here, can be envisioned as individual cells working in a complete distinction from their neighbors. Unitary smooth muscle should be envisioned as a system where the the smooth muscle cells work in a single unit. That is, if a muscle cell in this system contracts, all its neighbors will contract as well. Due to the fact that these guys are found in the wall of most of our viscera, that is hollow organs, some people, instead of using this unitary expression, they use visceral smooth muscle. You will also see this expression. This is why I call the attention. So, multi-unit smooth muscles, the cells will work independently, and therefore each cell receives its own innervation. In this visceral or multi-unit smooth muscle cell, the cells are connected by gap junctions. Gap junctions that we have discussed in electric synapses. The innervation happens in varicosities, I will show you in a second, and not every cell receives its innervation, and this is what I wanted to mention. So this would be a theoretical multi-unit system with independent cells and each and every cell receiving its innervation, whereas this would be the visceral muscle with the cells connected by gap junctions, and certain cells don't get innervated. Rather, they would receive the stimulation coming via the gap junctions. Varicosities means that instead of having very clear synapse between the nerve and the cell here, the nerve just simply has these enlargements where it has the vesicles, and it releases the vesicles into this area, which then stimulate the smooth muscle cells. Please, yes? You might recall that a normal synapse, the axon ended very close to the cell, and there was a structural full of vesicles and then comes together again, runs, forms another big bowl with vesicles, runs again, another big bowl, and when the stimulation comes, the neurotransmitter is just released into the intracellular, extracellular environment surrounding these muscle cells. The reason why we have discussed sympathetic and parasympathetic innervation is because all these smooth muscle cells are innervated either by sympathetic or parasympathetic fibers, sometimes by both. This involves all your blood vessels, all your hollow organs, everything that where smooth muscle is found, sympathetic or parasympathetic innervation, and that those particular neurotransmitters that I discussed are involved. So, my task now is very simple. You know the steps in excitation, contraction, coupling. All I need to say that the steps in skeletal and smooth muscle are essentially the same. It's just that how do they look is different. So we will discuss the action potential, changes in calcium, thin and thick filament activation in order for you to understand smooth muscle physiology. So first, action potentials. Well, on the right there you see a skeletal muscle action potential with the contraction as shown in the previous lecture. This guy shows you the same for the heart. This will be discussed 2 weeks from now in the second. I just put it here for completion. I won't talk about it. And down here you see the smooth muscle action. Please bear with me that when I hesitate in saying a smooth muscle action potential or the smooth muscle action potential, I have to say a smooth muscle action potential. This will be clear in the next slide. Okay, so some similarities and some differences. Well, obviously you see an upstroke and a repolarization, so first you might not be so much surprised that this is an action potential. However, please note the resting of the potential. As compared to striated muscle, smooth muscles have a much depolarized resting membrane potential. Consequently, or if you wish, the peak of the action potential barely goes above 0. Please note the time scale. The action potentials in smooth muscle are much lower than in skeletal muscle. Please also note the latency between contraction here, which is a couple of 100 milliseconds, or the latency of contraction here, which is a couple of milliseconds. So there are obvious differences and there are some similarities, of course. So I just took a few examples from different smooth muscle cells in our body to show you that one cannot say that this is the smooth muscle action potential. One can say that this is a smooth muscle action potential. Even within the same organ. This is your stomach and certain smooth muscle cells within the stomach. You can see that the membrane potential, resting membrane potential is different. The size of the amplitude and some slight characteristics are different even inside the taken from the urinary bladder. This one down here is taken from, from your small intestine. So smooth muscle cells produce all sorts of different action potentials. Some look like spikes, but they are very long. Nevertheless, the statement that this is sodium and this is potassium remains true. It's just that the channels are different and they have a different kinetics, therefore. Some action potentials have a plateau phase, like this guy here, or that guy there. Usually, for those cases where long contractions are needed, stomach and the gallbladder to empty, and the plateau is the consequence of calcium ions entering the cell. So sodium, calcium, and potassium ions are responsible for these guys. Sometimes you can see even these slow fades. This is the characteristic of the small intestine, where changes in the activity of the sodium-potassium pump can be so great that the changes in the resting membrane potential can be observed on the scale of tens of seconds. So, smooth muscle action potential is concluded by saying that it's a wide variety with some similarities to what we have already seen. Contractile proteins. Well, first, obviously, there is no such thing as a sarcomere since there are no striations. So what would be the analogous to a sarcomere? Well, if you look more closely on a smooth muscle cell, you will be able to find thin filaments and thick filaments as well. However, thick filaments are starting out from little rounded shaped things, which we call dense bodies. If you look at the composition of a thin and a thick filament, then not much is changed. However, please note the fact that in smooth muscle, although actin and tropomyosin are present, there is no such thing as troponin. A thick filament is composed of myosin. So this, the arrangement of the thin filament, the arrangement of the myosin in its head looks exactly like that in skeletal muscle. So the question then comes, if there is no troponin, what regulates smooth muscle contraction? Well, ladies and gentlemen, system is made such that students have to learn little. So the actomyosin cycle that I have shown you for striated muscle is the same in smooth muscle. It's only the activation of the system that is different. What is shown here is that in order for the myosin or the system to produce the ATPase activity, that is the hydrolysis of ATP, myosin needs to be phosphorylated. So what happens is that calcium in the intracellular environment of the smooth muscle cell increases, then it binds to a molecule called calmodulin. Has this molecule been known to you before? Very good. So this calcium calmodulin complex then activates a kinase, the calcium calmodulin dependent kinase. The COM kinase that has been mentioned in such a way. If not, don't worry. This kinase then goes to the myosin. It has a regulatory light chain and phosphorylates this regulatory light chain. So due to the biochemists having such a good way of naming things. This kinase can be called the calcium co-module-independent myosin light chain kinase. If one learns the name, one knows the function. So again, calcium co-module-independent myosin light chain kinase. Okay, so the myosin light chain gets phosphorylated, and then, and only then, can the up-to-myosin interaction take place. Okay? Clear to everyone? This cycle then goes as it does for the skeletal muscle that I have described. However, please note that in skeletal or striated muscle, removing of calcium would stop the contraction. Here, removing the calcium will not stop it because the myosin is already phosphorylated, the myosin regulated light chain is already phosphorylated. So in order to stop the contraction, you need to dephosphorylate the myosin. So there are phosphatases to dephosphorylate the myosin and to stop the contraction. Okay? So contraction is initiated by phosphorylation of the myosin. Relaxation is initiated by dephosphorylation of the myosin, and this balance is kept. You would then readily ask the question that in striated muscle, the regulation was based on thin filament changes. In smooth muscle, this previous thing was based on thick filament regulation. Does the thin filament in any way regulate smooth muscle contraction? Well, the answer is yes. There are a number of molecules that regulate thin filament, through the thin filament, the contraction, and I only took one component to describe you this system. But before I explain what calponin and all these molecules on the thin filament do, I would like to make you understand that smooth muscle regulation, that is through the thin filament, is very essential in your life and has to be completely different from that in striated muscle. And also would like to explain you why smooth muscle cells cannot have striations. Well, please remember what I have said, and everyone here agreed with me, that if you take the muscle and start pulling at the end, then you can pull the thin filaments out from underneath the thick filaments, and then no contraction can anymore occur. So let's take the theoretical experiment of doing the same with smooth muscle. Okay? That is, let's assume that smooth muscle would work the same way and see what would happen. Well, imagine the case, which never happens, of course, that a student has a lecture at 8 o'clock in the morning and wakes up at 7:50. And this never happens. Students usually wake up at 6 to be sure to arrive at 8 at the lecture. Yes? So this is an absolutely theoretical experiment that we are talking about. So the student wakes up at 7:50 and realizes that he would like to be present at the lecture at 8. So puts clothes on and runs to the lecture, and does not have the time to go out, if you understand what I mean, where the person didn't have time to go out. So the student sits in the lecture and would like to go out very much. Very much. And then the lecturer takes the absolutely unexpected route of not giving a recess between the first and the second So the student cannot go out. So what happens? If the smooth muscle cell would work like your striated muscle, that is stretching and stretching and stretching, and after being stretched, it cannot contract. Well, after a while, the student can't go out at all. And that would be disastrous. I hope you all understand. So, stretching our smooth muscle cells cannot result in a change of how they can contract. Nevertheless, you need some regulation of how many thick and thin filaments can attach to one another, and therefore regulate the number of cross bridges. Now this is done in the smooth muscle cell by having molecules on the thin filament. So now I come back to calcitonin and explain what happens. These guys, and from now on I will just say calcitonin. So the binding of calcitonin to the thin filament depends on its state of phosphorylation. If calcitonin is not phosphorylated, it binds to the thin filament and prevents myosin heads from binding. If calcitonin is phosphorylated, then it dissociates from the thin filaments and myosin heads can bind. Of course, myosin head needs to be phosphorylated in order to bind, but that we have already discussed. So the number of possible cross bridges that are formed depends on the phosphorylation state of calcitonin. Therefore, the number of calcitonin molecules being bound to the thin filament regulates the number of possible cross bridges that can occur under that particular state. And therefore, just like in skeletal muscle, you can and you do regulate the number of cross bridges that can at a certain time be formed, and therefore you can regulate the possible maximal force that you can produce. However, this does not depend on the length of the smooth muscle. Okay? Clear to everyone? Very good. There's one more thing that is very essential to be known and realized in smooth muscle regulation of smooth muscle contraction. That is the presence of phosphatases. As I have said, the possibility of a myosin head to attach to actin and therefore produce force depends on its phosphorylation state. So in skeletal muscle, contraction is brought about by changes in calcium concentration. At first, one would think that in smooth muscle, due to the fact that myosin head phosphorylation also depends on calcium, smooth muscle contraction and relaxation closely follows changes in calcium concentration. But that's not the case, and this is what I would like to explain you why. This shows the tension as a function of calcium concentration. If phosphatases are not activated, and this shows you the tension that is generated if phosphatases are activated. So what happens if you take an intermediate calcium concentration, let's say something here. If the phosphatases are not activated, system going from this calcium concentration to this calcium concentration, it will contract. Okay. However, if at this point calcium doesn't change, but something activates phosphatases, the smooth muscle cell will relax. stop phosphatase activity, no change in calcium, the system will contract. Relax and contract. So the regulation of smooth muscle contraction can be brought about by simply regulating the activity of phosphatases. Please note that, for example, a beta adrenergic stimulation, this will become clear when things discuss the smooth muscles of the blood vessels, and the increases in cyclic AMP actually does this. Activates phosphatases and relaxes the smooth muscle without anything to be done with calcium. Okay, so again, the tension versus calcium function shifts as the phosphatase is being activated or stopped. Finally, I would also like to say a few words about the smooth muscle energetics using the example that I have before. Under normal conditions, if you stretch a smooth muscle, the smooth muscle will contract. Please note that, for example, taking the previous example, when the student finally has the possibility to go out, it is the stretch that results in the contraction and therefore the emptying of the bladder. Optimizing interaction for each and every cycle requires ATP. So when the student is sitting in here and hasn't been able to go out and feels the contraction of the bladder, this contraction under, if everything would work as I have said before, and haven't explained what is on the slide yet, you would be using ATP continuously because your bladder is contracted. You're not going out because of having learned that you can hold certain things back, but the contraction is still there. So smooth muscle cells can maintain a contracted state for long times. This would, in theory, use a lot of ATP, and if nothing would happen, ladies and gentlemen, you would be eating fat 24/7 in order to produce enough ATP for these contractions. So how can smooth muscle preserve the contractile state without using much of energy? Well, actually it can, and we call this the so-called latch state. Latch is simply explained as the fact that the force is maintained, whereas ATP consumption is reduced. So how is that brought about? Well, as we discussed, the optimizing cycle to go on, you need the phosphorylation of the myosin light chain, regulatory light chain, so you have the phosphoryl. But what happens if the myosin and actin are bound and you dephosphorylate the myosin? Well, the actin and the myosin remain bound, still generate the force, but the cycle cannot go on. So the force is maintained, but ATP is not used. Let me try to give you an example that you might have heard of. What happens if the system in skeletal muscle gets to this state? When can that happen in a skeletal muscle? Well, in order for actin and myosin to remain bound and not detached. The detachment, if you recall, depends on the presence of ATP. So if this can only remain attached if there is no ATP. However, in order for myosin to be able to bind to actin, the tropomyosin and troponin needs to move away. So for that, you need the presence of calcium. So this state in a skeletal muscle can only occur if there is no ATP, but there is calcium. Can someone guess what this state is?
Yeah.
Yes. This is when the cell has died. No ATP, calcium enters. What happens to a skeletal muscle after death? Do you know the state that it is in? It's rigor mortis. The system is in a stiff, very strongly attached state. So in our skeletal muscle, this state does not occur under physiological conditions, but if it occurs under certain conditions, it does produce a very strong force. Not until proteases sort of degrade the proteins, the contractile spin filament proteins, will rigor mortis go away. You know, you can, in forensic medicine, from the temperature, very clearly guess, and from the state of the rigor mortis, very clearly guess when did death occur. You have probably seen what CSI or NCIS or whatever films where they say that, oh, the person died at between 11 and 11:15. So coming back to smooth muscle, this latch. You know the word latch? It's a way of a lock, actually, something that locks and holds in place. So this latch state is a way of keeping up force without consuming much of ATP. Now, due to the fact what I have just described, that is, even if you stretch the smooth muscle, it will adapt to this new length and won't alter the possibility of it contracting. As together with the fact that even if it is activated, it can stay in this activated state for a very long time. Smooth muscles have no real resting length. You cannot assign a certain specific length to your smooth muscle because it adapts to whatever it has to hold. Imagine the change of the uterine smooth muscles during pregnancy. So what is its resting length? What it was at month 1 or month 7. It's resting in every case. Okay? So this capability or this thing of the smooth muscle is called plasticity. Finally, just for a small overview, I would like to mention a number of things that affect smooth muscle regulation. Please note that at this point you do not need to know these, but as lectures go on and along your studies in physiology, you will encounter all these that are listed. There are a number of tissue factors that can influence smooth muscle contraction, usually things that are associated with metabolism. There are a number of factors that circulate in your blood or come from nerves that can affect your smooth muscle contraction, from acetylcholine and norepinephrine to other isoactive polypeptides to form with hormones and so on. And all these you will need to understand